So good evening, everybody, and welcome back to BrisScience for 2021. We are back. We're starting again online, but we are hoping to be back in person in the not too distant future. But for now, it makes it even easier for all of you to get into some science from the comfort of your own homes. If this is your first BrisScience, welcome. We are a free public lecture series on science where we aim to bring not just the best scientists, but also the best communicators to share their latest research with the audiences of Brisbane and now the world. We're hosted by the University of Queensland, our long supporters, and I'm your MC, Joel Gilmore. Great to be here. I'd like to start by acknowledging the traditional owners of the many lands on which we're meeting tonight and pay my respects to Aboriginal elders, both past and present. I'd also like to acknowledge those whose ongoing efforts to protect and promote Aboriginal and Torres Strait Islander culture will leave a lasting legacy for future elders and leaders. And I'd also want to call out today all the fantastic women in science, because it is International Women's Day today. So thank you for all your fantastic contributions, and also to recognise that there is a lot more work that we need to do in science and beyond to achieve true gender equality across our fields. So just a reminder, there is lots more to do there. So tonight we are, as I said, online using the Zoom platform. So the evening is going to run. We're going to hear from our fantastic speaker in a moment. They're going to present, and then we'll have the opportunity for some questions and answers at the end. You can do that using the Q&A function in the app. There's also a chat function if you need to talk to us, the organisers, about anything. But if you've got questions, pop them in the Q&A, and at the end of the evening, we'll go through as many of those as we can. This session will be recorded, but your names will not be used, or maybe I'll use your first name if it's in the chat, and then you can catch up on this along with all the other BrisScience talks on YouTube afterwards. So tonight we are diving back into everybody's favourite topic, which is, of course, pandemics. We're being joined tonight by Professor Nigel Perkins, who is the head of the School of Veterinary Science at the University of Queensland. Now, why a veterinary science speaker? Well, Nigel is a zoonotic disease expert, as in animal to human diseases. And his talk tonight is ominously titled Preparing for the Big One, which sort of felt to me like what the last 12 months were about. But so I'm a little bit nervous about what's coming up next. However, in addition to his work with the World Health Organization and his many roles at UQ, Nigel is an award-winning, let's try that again, a multi-award-winning teacher. So tonight we are in very good, very well disinfected hands. So please join me in welcoming Professor Nigel Perkins.
Thanks, Joel. I will just share my screen and to everyone. Look, thanks very much for the wonderful introduction. It's a little bit unusual. This is the first BrisScience event, so it's exciting to be here in front of you. A little bit unusual because I can't see you all, but I can see that there's a large number of people attending as participants. So fantastic to be here. As Joel said, I'm a veterinarian. I will be talking primarily about the origins of pandemic risk, and starting with wildlife and animal reservoir hosts and heading up to human exposure and infection. I must provide a little bit of a disclaimer at the beginning. I'm aware that there may be a lot of interest in the audience about human dimensions of pandemic disease, and my disclaimer is that as a veterinarian, it's less appropriate for me to be commenting too much about human therapeutics and diagnostics and so on. So we'll be focusing mainly about zoonoses and preparing for the next big one, potentially, accepting the comment that we may already be we certainly are in one of the big ones, but perhaps not, it could be worse than it is, and the next one may be worse than the current one. So what do we mean about zoonoses? The World Health Organization describes zoonoses as diseases of animals that are naturally transmissible to humans, and that distinction will become a little bit more important in a minute or two. There are many, many zoonoses active around the world at any point in time, perhaps as many as 200. And they include all of the types of pathogens that we might all be familiar with: viruses, bacteria, fungi, prion diseases, these rather unusual entities that are capable of causing transmissible spongiform encephalopathies, so mad cow disease, for example, and parasites. Tonight, we'll be talking mainly about viral diseases, largely because of the current pandemic. But there are many other causes of zoonotic diseases. The World Health Organization maintains a list of priority blueprint or a blueprint of priority diseases with pandemic potential. And many of these are zoonotic. And this is, this is where the sort of term, what is the scary big one? At the moment, we're in the midst of the SARS-CoV-2 pandemic. Perhaps some of the influenza virus, the risk, the Spanish flu from 1918, 1919, and some of the bird flu subtypes that have been circulating around the world or in different parts of the world since the late 1990s. So the H5N1 bird flu originated human diseases that occurred in Hong Kong and elsewhere in Asia. And then since then, H5N2 and H5N6, these are all bird flu derived human cases of influenza virus. And some of these have a much higher mortality rate than the current SARS-CoV-2 pandemic. And so perhaps even more scary potentially than the current pandemic that we find ourselves in. And there are various influenza subtypes, colloquially called swine flu subtypes. And these are typified by the swine flu pandemic that occurred in 2009. And that involved a flu virus that was probably a combination of 4 different virus segments, from swine origin influenza virus, bird influenza viruses, and human influenza viruses. Down the bottom there, you'll see disease X, and the World Health Organization includes this unknown disease. So disease X could be the next big one. Disease X is a future disease that is not yet discovered or characterized, and that might have the 2 big drivers of the big one, and those are transmissibility or reproductive rate. It's amazing how much epidemiology and zoonotic disease terminology we have become accustomed to through the last 12 months. So the 2 big issues that drive the big one is transmissibility, so the ability to spread from human to human, and the other is virulence or severity of disease. And those are the things that make the scary diseases. So the big one of the future is something that spreads as rapidly as the current SARS-CoV-2 virus, but perhaps has an even higher mortality rate. Global outbreaks are increasing over time, and we'll talk a little bit about the drivers that might explain that. And within global outbreaks, so these are global outbreaks of human infectious diseases. And animal origin diseases are accounting for an increasing proportion of global outbreaks over time. So up to 75% of new diseases or emerging infectious diseases of people are thought to have come from an animal origin. And collectively, all of these diseases account for large numbers of hospitalizations and serious disease in people around the world, and large numbers of deaths. There's a huge impact of zoonotic diseases on people every year. And as we'll go through in this presentation, controlling or eradicating zoonotic disease or preventing human risk is probably best pointed at the animal and wildlife interface. It's important to understand a little bit of the distinction here, and this is a bit of a complex graphic. The top bar talks about zoonoses, and a classic zoonosis, this is where I come back to the definition that I talked about right on the first slide or the second slide. The zoonoses are diseases of animals that are naturally transmissible to humans. And so the classic zoonosis is a disease that every time a human gets infected, it requires the animal origin or the animal reservoir host to be a part of that infection or exposure piece. And so rabies is the classical example. 60,000 or more deaths still occur per year due to rabies, and 99% of human cases come from dog bites. So that's a classic zoonosis where there is an animal reservoir host involved in almost every human infection. Most of those, so 99% of the classic zoonoses, come from domestic animals. So the reservoir hosts are domestic animals, and these include many of the foodborne diseases that affect people. And just 1% come from wildlife. In contrast, we have this second group here, which is the emerging infectious diseases of animal origin. So these are diseases that have an animal origin, but are now capable of independently being transmitted from human to human. So they no longer require separate animal exposure events because they're spreading from human to human. And this is where influenza or bird flu origin and SARS and COVID-19 sit. They certainly came from an animal origin, and there could still be animal exposure events happening, but the vast majority of human cases and human disease is now occurring independent of any further animal transmission to humans. It's all associated with human to human. And notice that most of these emerging infectious diseases of animal origin come from wildlife. So 72% of them are directly associated from wildlife. If we look at the biomass of the mammalian biomass around the world from which most of these diseases originate, wildlife account for less than 1% of the total mammalian biomass. Humans account for about 36%, and the rest is largely terrestrial animals and marine mammals. We now know with reasonable confidence, with fairly high confidence, that bats, particularly horseshoe bats in China, are the reservoir host for SARS-CoV-2, the virus that is associated with COVID-19. And that this virus was likely to have moved from a bat to an intermediate animal host before infecting humans. And beyond that, the rest is a little bit uncertain. We know that the SARS-CoV-2 virus doesn't replicate well in other domestic animal species, particularly pigs, chickens, and ducks. And there's a lot of conjecture about the intermediate animal host. Is it the pangolin, this rather unusual animal? Could it be a snake or some combination of the two? And we'll talk a little bit more about this, and in the end, argue that it probably doesn't matter too much. Pangolins certainly are implicated, but there could be other animal species as well. It's of great interest to note that SARS-CoV-2 goes from humans to some domestic animal species. So cats and dogs, ferrets, mink, and other large species of cats have all been thought to have contracted SARS-CoV-2 infection from humans. And there's still considerable uncertainty about whether those domestic animal species and the large wild cats can in turn then transfer virus or cross-infect other people. There's very few cases of these around the world of domestic animals and large cats, and there just isn't enough evidence of subsequent human infection. Is it all about bats? There are many, many coronaviruses circulating in bat species, and this phylogenetic tree just shows the relatedness. All of those red isolates are different human isolates of SARS-CoV-2 virus. And then adjacent to that, so the most closely adjacent clades are the most related in a genomic or phenotypic sense. So you can see down here where it says bat SL, that's a bat SARS-like coronavirus, not identical to the SARS-CoV-2 virus, but very similar. So bat sampling in China has isolated coronaviruses from these little horseshoe bats that have a very high genomic similarity to human isolates, 96%. 96% sounds like it's really similar, but that 4% divergence could take decades or could take many years to occur, given the natural rates of mutation and recombination. Bat viruses have different receptor binding domains or spike proteins. Again, isn't it amazing how familiar we are with some of these terms? Bat coronaviruses do not normally infect human cell lines. They're not capable of infecting human cells directly because they have a different receptor binding domain. The viruses are different. Pangolin virus isolates of coronavirus have been shown to infect human cell lines. So again, that's a bit of information to implicate pangolins in SARS-CoV-2. This little graphic, just as an update, I'm trying to use graphics from fairly recent and current papers, and where possible, I'll give the citation down on the slide. So for those of you that are interested, if you download and watch the recording, you'll be able to follow this up and chase up the articles and read in a bit more for a bit more depth. This graphic just shows a couple of the pathways that could be involved in shifting the virus from bats to humans. One involves bat coronaviruses undergoing mutation and or recombination, so undergoing adaptive change inside an animal host. And the pangolin there shows an animal that has been infected with multiple separate viruses at the same time that could be from human origin, other animal origin, and bat origin. And when you think about mutation and recombination, RNA viruses are able to recombine or take genetic segments from other viruses in the same infected animal host and put them together into a new virus strain that can then be released. So it's unclear whether pangolins were the intermediate host, and the animal in the middle with the question mark on it, this animal here, is an unknown animal species that could have been an alternate intermediate host. So the most likely explanation is a bat virus going through an intermediate animal host that then acquires through mutation or recombination the ability to bind and infect human cells and then subsequently human exposure. There's a slim possibility that this could have occurred through direct transfer of bat viruses to humans. But in the bat virus isolates that have been found to date, there's enough difference to suggest that this pathway probably didn't happen, or probably hasn't happened today. It's important to understand a little bit about cross-species infection with viruses. There's an enormous number of protein interactions that have to occur for a virus to successfully attach to, enter a human cell, and then undergo viral replication and then be shed. So successful infection involves somewhere between 100 to 300 separate protein-based interactions, and errors at any one of these mean the difference between a dead end or non-infection or a dead end host versus successful infection and replication. So most cross-species infections probably never go anywhere. Over time, selection pressure leads to the occasional cross-species success, where a virus infects a different species of animal or a human, and then very rarely goes through the whole business of infecting, replicating, and shedding. And so this leads to the commonalities in here, the fact that these receptor binding domains, for example, might be common across different human cells. It leads to different viruses slowly adapting similar strategies. So different viruses may develop similar receptor binding domains to allow them to infect human cells. Coronaviruses and influenza viruses and a number of the other zoonotic viruses with pandemic potential are single-stranded RNA viruses. And this is a particular type of virus. So single-stranded RNA viruses have a relatively small genome, and they undergo very rapid replications, and there are very large populations of RNA viruses, so there's lots of virus diversity. High rates of mutation, high rates of recombination, and there's a distinction between recombination, and that's the little graphic there on the right, recombination where base pairs might be adopted by other genes and it results in a small level of antigenic change in a virus, and reassortment where entire genes might be shared between different viruses. So many mutations are non-survivable. Some lead to adaptive advantage, and adaptive advantage simply means that in an evolutionary timescale, those viruses with that adaptive advantage are more likely to survive and replicate, and therefore will become the predominant strain. And this little graphic down the left, again, a little bit complicated. It just shows that for these RNA viruses, as they undergo replication, there will be lots of virus diversity. And so that's up here, this idea that there are large numbers of different viruses and they're undergoing rapid replication with very high rates of mutation. So lots of diverse viruses in the same individual. There's a combination of mutation, which is individual base pairs or very tiny segments, and recombination, where larger segments get shared. When mutation rates are really high, there's likely to be a very high error rate and those viruses will be unsuccessful. And there's a surface here where the combination, where recombination plus mutations lead to this maximal adaptive change. And these are viruses that change very frequently. So RNA viruses are under constant selection pressure, and they have to be, because they've got to evade all of the immune responses that any animal or human is capable of bringing to bear. This creates opportunity to infect the same host over time, and this is why we need a seasonal influenza vaccine every year to account for mutation in our regular seasonal flu. And it also allows RNA viruses to be able to jump from species to species to successfully manage spillover infection. We can think of this as a massive exercise in mathematical probability or chance. If you were to undergo the same repetitive thing time after time after time, you can expect a small likelihood of success. So imagine a hole in one. Even if you're not a successful golfer, if you were to hit shots on a par 3 hole thousands and thousands of times over many years, you can expect to get some holes in one. That's the kind of mathematical exercise that we're talking about. And if you run that out to a lifetime, so you can see that the odds of an individual stroke might be vanishingly small. The odds of successfully getting a hole in one, even with 1,000 strokes, might be still pretty small. But if you manage that out over 25 rounds a year for 40 years, the probability of getting at least one hole in one becomes an appreciable number or proportion, 20% chance of getting at least one hole in one over a 40-year period. That's the kind of rare event that we're talking about with cross-species spillover events. Remember, we said very large populations of RNA viruses that are undergoing rapid mutations and recombinations. Most of them die, but occasionally there will be a success just due to random error. And if we then add some large-scale drivers that increase the probability of those events occurring, well, we shift the likelihood a little bit towards a higher probability. And these are the most common drivers of emerging infectious disease outbreaks. Many of them associated with poor public health response capacity, and that's really a marker of developing countries that have a lack of infrastructure, and a lack of training, and a lack of public health capacity. But a lot of these drivers, so the numbers around the outside of this pie graph is a representation of the number of EID events summarised in this particular paper. Many of these changes are associated with land use and agricultural practices. So you've got food and agriculture, climate change, and local weather events, international travel and commerce, a whole bunch of drivers there that increase the likelihood of spillover events. We can appreciate these visually, and these are the global trends that are driving this zoonotic disease risk and driving the increased risk for emerging infectious diseases. We have huge population growth and urbanisation. We've got massive connectedness and rapid travel of animals and animal products and people around the globe. We have environmental disruption on scales that have never occurred before, and much of this is all about commerce and planting food or raising food to meet our increasing population demands. And intensive agriculture is often monoculture driven, so large crops of the same, large acreages of the same crop, large numbers of animals all housed together. That increases the likelihood of an outbreak if an infectious disease were to enter that particular environment. Down on the left there is the expanding or the enabling effect of antimicrobial resistance and perhaps inopportune use of antimicrobials in animals and in humans. And over here on the right is some of these unusual circumstances that we think contribute to increased wildlife to human spillover, and particularly in some developing countries where different animal species are housed together and in close proximity to people. And these are some of the particular examples. So this is SARS, the original SARS outbreak due to coronavirus that began in southern China in 2002, 2003. And these are photographs that I took in a restaurant in Guangzhou. So that's in southern China, and this is just outside this landmark restaurant. And we can see animal cages and then seafood cages where a lot of live animals are displayed, and the patrons can choose their food and then point those out to the restaurant staff, and those animals then get butchered on site and cooked and delivered. And this is a closer up photograph of this exact same photo here. And so you can see in here that there's chickens and ducks, waterfowl, there's cats and dogs. Down here, this photo, so in this intermediate cage, this photo here is a blow up of that scene there. So this is a palm civet sitting underneath a snake, which in turn is sitting underneath waterfowl and domestic poultry and cats and dogs. And this is the table that I was sitting at inside that restaurant a little bit later. And in SARS, in wildlife and wet markets in southern China, SARS virus was found in palm civets, but also in these other wildlife species, both raccoon dogs and ferret badgers. And this is the Wuhan wildlife market that is postulated to be the source of SARS-CoV-2 in the current COVID outbreak. And this is a very large commercial or industrial scale wildlife and seafood and fruit and vegetable market that serves a large population in southern China. It's not clear whether there were pangolins in that market, but there were certainly many wildlife species, and many of these wildlife species, it's even a little bit of a misnomer to describe them as wild animals. So bamboo rats and civets, for example, these are wild species of animal, but because of the demand for this meat, these animals are farmed very intensively, and so they're really treated more like domesticated farmed animals. I really like this graphic. It shows the spillover risk at two different interfaces, and it talks about where we might affect interventions to try and prevent spillover risk. So this graphic up here is uncontrolled risk, and initially we can see virus circulating in a wildlife reservoir. So that's what this little up and down pattern is. Every now and then there's a spillover event from wildlife species into a domestic animal species or an amplifying host, and that increases. Amplifying hosts act to increase the amount of virus in the environment. And then we can have a spillover effect either from the domestic amplifying host or from a wildlife host into people, and we get a human outbreak. And the point of early detection and intervention is to try and dampen both the domestic animal case surge and the human case surge. And in general, we want to be trying to intervene to the left on this curve, so further ahead of the infection of the chain of infection relative to human exposure. The best place to be intervening is way out here in the wildlife and wild environment phase to try and stop the risk of spillover to domestic animals. It's also useful to try and intervene in the domestic animal amplifying host phase to try and prevent amplification and reduce risk to people. The least effective control phase is to be intervening once there is already human-to-human transmission and an emerging pandemic or epidemic. But often that's where we find ourselves. And that's the purpose of this graphic. The idea, again, is that we want to be intervening way down here at the interface between wildlife and an index case, the spillover that results in the very first human infection. Often we end up not intervening until we have multinational or pandemic spread. So the further back to the left towards the origin of zoonotic diseases is where we're going to have the largest impact. That's why people like myself, veterinarians, are fascinated by this whole exercise. There's an enormous amount of work that's gone on to try and identify parts of the world where novel infectious diseases and particularly viruses might be most likely to emerge. And this is the heat map where the different colours represent increasing probability of zoonotic viruses. So the probability that a sample would find an emerging infectious disease. And you can see that much of the highest risk is in the tropics, and it varies for different types of wildlife and it varies for different types of viruses. There are many different versions or different studies that have looked at different attributes. In general, those animal species, so zoonotic viruses are more likely to be found where there are host species, so wildlife species sharing tropical forests with large amounts of biodiversity and where there's opportunity for ecological contact, so opportunity for these host species to contact domestic animals and humans. in recent years, there's been quite a lot of work to try and figure out whether, is there something special about bats? Do bats carry more zoonotic virus than other species? And this paper published just last year suggests that the number of species of an order drives the number of viruses. And rodents and bats account for a large percentage of total mammalian species. So rodents alone account for 40% of all mammalian species. So they have a huge species richness. Bats also account for a large proportion of mammalian species. Bats account for 25% of all mammalian species. This paper has shown that the number of species in an order, so for example, a number of species inside bats drives the number of viruses, and the number of viruses in turn drives the number of zoonotic viruses. So on this plot here, we have on the x-axis, it's the number of virus species in a particular type of animal. And on the y-axis, the number of zoonotic viral species. So you can see they're very directly correlated. And so it's not about anything terribly different about bats. It's just that there are lots of bats, very diverse, and lots of bats mean lots of viruses. And it's lots of viruses that drive the risk of zoonotic viruses. There is a lot of interest in the types of viruses that different animals carry and increased phylogenetic distance. So the more genetic difference there is between an animal species for the wildlife host and humans, it produces zoonotic viral diseases that may be less transmissible and more highly fatal. So bats are further phylogenetically from humans than primates, for example. So bat-borne zoonotic viruses are likely to be less transmissible in humans, but cause more severe disease. There's been a lot of work in the last decade or so about using high-tech solutions to manage prediction and prevention of human zoonotic disease risk. And this is just one of several papers that describe the approaches to this kind of activity. It starts up here, a series of steps in here. So the first, labelled A, up in this top left corner, is all about detection and discovery of what viruses are out there in wildlife and in the environment. Pathogen discovery and developing diagnostic tests to allow us to discover where viruses are. This section over here is the use of fancy and integrated data analytics, so data sources, rapid diagnostic testing, so using the information out of diagnostic tests to try and predict where zoonotic viruses are likely to occur. And that's really the map that I just showed you a minute or two ago. We can use all of that information together to look at early detection and real-time forecasting. If we know where in the world zoonotic disease outbreaks are likely to occur and we have really good diagnostics to try and detect them, then we could invest a lot of effort in those areas that are trying to detect outbreaks very early in the chain of infection, down around emergence or where there's very few human cases, and then be prepared for improved response capacity. That's really nice, and that's using big data and lots of high capacity and high tech to produce some predictions. There's lots of international agencies that are active in this space, and the big three in the international aid agency space are the World Health Organization, responsible for human health international efforts, the OIE, or the World Animal Health Organization, responsible for animal health, and FAO, responsible for a lot of animal production and overlapping with OIE in the animal health space. These three agencies collaborate tremendously in zoonotic disease and One Health areas, and together they support the global early warning system that is aimed at detecting infectious diseases as they emerge at the urban wildlife interface. There are many other agencies that are active in this space, and one of the big ones is the USAID program called the Emerging Pandemic Threats Program, and it has these 4 component projects: predict, prevent, identify, and respond. And those overlap with the graphic that I just talked about on the previous slide. There's a huge amount of really high-tech stuff that's been going on in the last several years, and these are 3 really big examples of what I would call moonshot science, and they don't come much bigger than these projects. The Global Virome Project, is aiming to characterize all of the planet's threatening viruses and assess the spillover risk based on their genomic sequencing. So every virus in the world from any animal or animal source. The BioScan project has gone even bigger. Globally accessible DNA systems to describe all multicellular life on the planet, and the Earth Biogenome Project, complete genome sequences for every plant, animal, and fungal species on the planet. It is mind-boggling to think of the scale of these activities. And the idea here is to use big science and big data to prepare for the big one before it can jump on us. There's a fair bit of skepticism around the world about whether this is the right approach and whether this will help prepare us for the next big one, but it is probably a useful and necessary component. There's a growing emphasis towards activities that focus at the other end of the spectrum, and this is really the area that excites me. The focus on high-tech solutions is probably not enough. Black box, big data complexity, which is science at its best, I will admit, and it is inspiring and it's visionary and inspirational. But the real challenges, the wicked policy and the implementation challenges are down at the local scale. The local narrative is a key. These are local contextual norms. These are social, cultural, religious, political, and other norms that drive human behaviour at the local scale. And this is intimately linked to land use and animal use at the local scale. So land use induced spillover risk. This is about this cutting forests down and interfering at the urban wildlife interface and human behaviour. So early detection and response capacity is driven by this local narrative. People have to be motivated to report unusual diseases, and there has to be some form of local health response capacity to investigate those if we're going to pick them up at emergence. And even better, if we want to implement control strategies that reduce this risk, we're influencing the same human behaviours at the local level. These are the key. And there's a whole series of activity. This is a growing area for zoonotic disease management at source. And this is now considered to be the biggest driver of a future successful effort to reduce risk at the local interface. The high-tech solution is not going to be the big driver of successful prevention of the next big one. So in summary, I'm right at the end of the talk. We don't have the ability to predict the next pandemic with precision and accuracy. It's difficult to even detect emergence of spillover events before the epidemic stage. And to date, most of our big pandemic events have really reached international spread before we've detected them and been able to mount an effective response. Transdisciplinary collaboration is critical. Tech is great. Tech and big science is driving capability and response capacity, but local and country level management is the key to prevention, and we need to continue to put effort into these spaces. And this is the domain of One Health. One Health is this idea that we are connected, that the planet is a single unit, and everything we do inside this planet affects wildlife and animal health, it affects environmental health, and it affects human health. And our ability to optimize human health is dependent on improving the health of all. I just want to mention UQ, the last couple of slides, Joel. UQ is a global powerhouse in research, and it's got a huge capacity in human health research and emerging infectious diseases. The School of Veterinary Science lives within the Faculty of Science, and we are we're a minnow in the UQ powerhouse, but we love being here, and we are an important minnow. The School of Veterinary Science is involved in a large number of programs on zoonoses and emerging infectious diseases involving many of our faculty, and I just want to mention a couple. We're very active in antimicrobial resistance at the animal and environmental interface. We're very active in zoonotic diseases in Australia and elsewhere. Q fever is one example that I haven't been able to talk about. We're active across a broad range of zoonotic disease issues. And these are some examples that I won't go through in the interest of time, but you can see that some of these involve multinational efforts And some of them involve an Australian focus, for example, point of care rapid testing for hantavirus detection. And we're hugely excited to be a part of the Queensland Alliance for One Health Sciences, a multi-partnered, innovative and exciting venture that is addressing exactly the kinds of things that I've been talking about tonight. Transdisciplinary, big data, impact-oriented, and trying to have an impact at the local event, at the local scale, led by Ricardo Suarez Magales, one of the vet school's epidemiologists and zoonotic disease researchers. So lots of things we could do with more resources, but I'll end there, Joel, and be very happy to entertain some questions.
Thank you very much, Nigel. A fascinating talk and a little bit concerning as always, these things tend to be. We are going to go to questions in a moment. If you've got a question, pop it in the Q&A chat. I can see a few there. We've got time for a few questions. Just a reminder that you can catch this talk on YouTube in about next week. So don't miss that, as well as all the past BrisScience talks. And we'll be back next month online on the 12th of April. Speaker to be announced. You'll have to hold off just a little bit long before we reveal that. We'll look forward to seeing you then. All right, so Q&A. Nigel, going to jump in here. First question is from Chris, who asks, and if you know this, what was the purpose of the recent World Health Organization team visit to Wuhan and what was achieved?
Well, thanks, Chris. That's so the purpose ostensibly was to provide an expert panel, to allow an expert panel to visit what was considered to be the origin of COVID-19 and to understand that where the animal sources might, or what the animal sources might be, and the exposure pathways that led to the first or the early human cases. Lots of politics associated in that activity, and there is a preliminary report that's available to all, and I think there's a final report that we can expect in the near future.
Great, thank you. Got a question here from Kate, who notes that the solutions presented, or I think this might be actually the problems, that pie chart of problems, seem to align well with the climate movement, such as deforestation and mitigating climate change. Do you have any comments on that?
Yeah, thanks, Kate. It's true that climate change is driving, or climate change is having a major impact on a lot of the emerging infectious disease and zoonotic disease risk, and it does it through many different ways. It changes the distribution of vectors and animal host species for different diseases, and it's increasing perhaps the way that humans interact, increasing the likelihood of deforestation or the changes on the landscape. All of those things are associated with the kinds of drivers that I was talking about. So climate change is a recent addition to what has been fairly well described drivers for emerging infectious diseases over the last couple of decades. And it may well be that climate change has a huge impact going forward.
Thank you. Min asks, as the bat habitats have been affected by the bushfire in the last few years, many bats are coming to the residential areas in Brisbane. How can we prevent human and pets from getting viruses from bats and other intermediates such as rodents and reptiles? Is that a risk we should be worried about?
Thanks, Min. So in Australia, for example, there's been a lot of wonderful work done on understanding bat ecology and the changes in the bat ecology and bat landscape, or the environmental landscape that influences bat colony dynamics and subsequent impacts on disease spillover risk. Australian bats carry Australian bat lyssavirus, which is a rabies type virus capable of causing fatal disease in humans that come into direct contact with bats. So people in Australia should not be interacting with bats that are sick or damaged. They should be contacting wildlife carers. So that's associated with lyssavirus risk. Australian bats also carry hendrovirus, but there's very little evidence to suggest that hendrovirus can spill over directly from bats to humans. It mostly goes from bats to horses, and then horses act as the amplifying host and infect humans. So we have done the anthropomorphic effects, so human effects have have had big impacts on bat colony dynamics. We've fractured the bat landscape, so we have a larger number of smaller colonies that are more mobile and more dependent on human planted trees in the peri-urban environment for food, and that increases the likelihood of humans interacting with bats or with bats interacting with horses in the peri-urban environment. So lots of things that we can do to change that. That involves, I probably won't go into a lot of those details, but it involves tree planting, it involves management of horses to reduce spillover risk for Hendra, and it involves care and attention in how we interact with bats. I'll stop there, Joel.
Oh, thank you. And Tina, I hope that addresses your question as well in that question list. We are pretty much at time, but I'm going to run a couple minutes more because there are so many questions here. If you've got a few more minutes, Nigel.
Yep.
If anybody else needs to go, thank you so much for being here. We'll try and get through just a couple more questions. Got a question here from, it's from Anonymous, but why is Eastern Australia a global hotspot for potential zoonotic disease? I noticed that on the chart that it seemed to be a very high prominent spot as well, obviously being in the tropics, but is there anything that we're doing particularly problematic?
No, so that's a good question. It's likely to be bat-borne species. So it's likely to be the same drivers that I've described, but that bat-borne species tend to be more distributed across the eastern coast of Australia and then into the northwestern part, across the top of Australia and into the northwestern part of Australia. But it's the same drivers just acting on the local environment. We also have migratory birds that are coming in, so migratory water birds, for example, that carry some risk of introducing avian influenza viruses.
Right. All right, I'm going to ask last couple of questions. This one is just a fun one, so I'm really curious. Is there any truth to the stories that climate change is meaning humans are coming into contact with animals that have been frozen in the permafrost, which is now thawing and are getting diseases from them? I'm pretty sure that was an episode of House, but is this something we should be worried about?
Yeah, no, that's an interesting question, Joel. I'm not aware. So there's certainly in thawed, frozen animals and people, we can find genomic material that can be used to identify even infectious diseases or pathogens, if you like. So, for example, some of our sequence information, I understand, from the 1918 Spanish influenza outbreak, some of that sequence information came from people that had died in 1918 and had been buried in frozen tundra. So in those parts of the world where their bodies did not biodegrade. And so recent scientists were able to dig up some of those bodies with appropriate permission and isolate or not isolate, but gain access to remnant genomic material and sequence some of the virus. So I'm not sure that you can get live infected material, but you can certainly get genome material.
All right. Second last question, which is from Brent. Given the geographic knowledge of spillover risk, do you believe that governments from developed countries such as ours are doing enough to support the actions of the OIE and the WHO?
Yeah, really good question, Brent. It's easy to criticise a whole raft of different, or easy to discuss or criticise a whole raft of different aspects of pandemic prevention and indeed pandemic research. Some of the pandemic risk, or perhaps much of the pandemic risk that we face, is occurring in tropical areas, and many of those are associated with developing countries that might have poor infrastructure themselves. And so we end up with this exercise where we have high wealth individuals and entities and organisations and governments from Western developed countries attempting to do research in poor countries that has all kinds of political connotations and perceptions on it. And some of those overlap, or if you like, what would you call it, trickle over into the political domain. So in the early avian influenza outbreaks across Asia in the late 1990s and early 2000s, there were Asian countries that stopped foreign researchers coming in and getting access to isolates of human virus because of concerns about the politicization. the reality is that the next big one could occur in a developing country, in a poorer part of the world, but it may also originate in one of the Western countries. The drivers are indiscriminate, but it's in all of our interests to manage increased health capacity across the entire globe. And that's really the key message from my presentation. It's not big tech that is going to stop the next pandemic. Big tech will help us respond to it more quickly, and isn't that fantastic? But it is local collaborative involvement and local enablement, and that's all about social cultural issues that's going to prevent the next pandemic.
Well, look, I think actually that is a fantastic note to finish on, a bit of a call to action for us all. So thank you so much for your time this evening, Nigel. That has been informative and a tiny bit worrying for the future, but hopefully it's great to see so many different research groups at UQ and beyond preparing and making sure that if or when the next big one happens, we are really well prepared. So really appreciate your time tonight.
It's a pleasure. Thank you, everyone who's attended.
Right. Don't forget, there'll be a quick survey after you leave. So if you could fill that in, that'd be fantastic. Otherwise, we'll look forward to seeing you all next month. And again, happy Women's Day. Catch you all.